Hello everyone, welcome to our second video for the Blueprint of Life. We are looking at evidence for evolution. Just a quick note, this is a significantly big video, lots of content to get through here today for a single dot point, so let's just be aware of that. Alright, so this is our big dot point. We're asked to describe, using specific examples, how the theory of evolution is supported by the following areas of study: paleontology, biogeography, comparative embryology, comparative anatomy, and biochemistry. So the goal today is to have an understanding of how each of these supports evolution with examples. So there'll be a table right at the end that will help you lay this out for those people that don't want to spend the whole time writing notes. So I've kind of helped simplify that process for you all. Okay, let's get stuck into it. So, why do we need evidence? Well, we get evidence from experiments, which is great. We get a single little bit of evidence. This is awesome. We get more experiments, and they give us more evidence. So, when we start seeing a bunch of different evidence, we start to come up with a theory. And with the evidence that you have, you might come up with competing theories to explain what's going on with that evidence. As you conduct more experiments, you get more evidence, and you can discount some of the previous incorrect theories that were suggested. All these theories have some assumptions about them. However, we want to stick with the ones that make the fewest number of assumptions. Those are the ones that are more reliable. And so we have our theory here, and we create a picture from it. And while it may not be identical to the truth in the real world, we get there. So what we're looking at today is this idea that trying to dispel this concept that evolution is just a theory. Well, a theory is made out of body of evidence, and that evidence comes from these different regions. So that's how you should approach this video. We are trying to grab more dots to make sure that our understanding of evolution reflects the reality of the world and universe that we live in. Okay, so let's have a look at these 5 areas. The first is paleontology. This is the study of fossils. Biogeography. This is the study and distribution of species and ecosystems in geographic space and through geological time. So there's an element of understanding movement of tectonic plates and continents around the planet. The next one is comparative embryology. So we look at embryos in early stages of development. We have comparative anatomy. This is the study of similarities and differences in the anatomy or structures of different species. And then we have biochemistry, and that's the branch of science concerned with chemical processes and substances which occur inside a living organism. Alright, so we're going to break down each one of these things. So it should be you should be pretty comfortable with the formula that we're working through with this video. Alright, so looking at paleontology, it's really talking about fossils. So just remember that a fossil is the preserved remains of dead organisms. So over here we have the oldest fossil in this little picture of 570 years ago, and it's some sort of simple blobby type organism. And as we go up through the rocks get younger, and the organisms get more complex. So we've got some sort of turtle head, some sort of ape, maybe ape head, and then we have a primate head. So you can kind of see these, see the complexity increasing as these species get younger. So what this is actually what's called the fossil record, right? It's the ordered array in which fossils appear within the strata of sedimentary rock. So strata is the layers, and we know that old rocks are down the bottom, young rocks are up the top, and that's a good indication of showing that Firstly, organisms change. That's a good starting point to support evolution, is that evolution change over time or descent with modification. Right? And we also get that these species are not disjointed from each other, and we get them through transitional fossils or transitional forms. So these are fossils that specifically show the gradual transition from an ancient species to modern ones. We're going to have a look at a few of them now. So if we looked at the fossil remains and have a look at these, we've got a couple of missing bones here. So this is probably a good conversation to have right now, is that the fossil record's actually incomplete. We rarely have the conditions right for the formation of fossils. So the presence and record of these transitional forms are not always there. But anyway, so we've got a terrestrial Pachycetus. I'm not even bothering trying to say these names. But we've got a terrestrial organism. We've got that organism shifting into the water, predominantly aquatic. And then we have another one which is fully aquatic. And then we have our baleen whale. And this kind of shows you some sort of transition. But finding these transitional forms is really hard. So, we get there. Anyway, cool example of a recent discovery in 2016 was the early tetrapod. A tetrapod means tetra meaning four, pod meaning the legs that you stand on, and so it was a great example of a transitional form that linked a fish species into an organism that was able to leave the water and move up onto land. That was a great transitional form discovery to find. Okay, our next example, our next area of study is biogeography. So remembering this is the looking at the geographic distribution of species, and Darwin actually used this in his voyages, given that he got to see quite a bit of the planet. He saw that there were organisms sharing similarities, and he suggested that these all had a common ancestor. The example that we're going to use is one that we kind of should be already aware of because of our studies in year 11. It's the ratite bird. So the ratite birds were found on southern continents, and they share similarities with each other and the fossil evidence of an ancient Gondwanan bird. So these are our, there's a few more, but these are just a nice example of a selection of these ratites. So you've got the rhea, you've got the ostrich, the emu, the cassowary, and the kiwi, and they all have similarities with each other and with this ancient bird. So this suggests a common ancestor. The ancient bird was the common ancestor for all of them. Alright, we have comparative anatomy. Alright, so we are looking at the structures of the body, or bodies of organisms. Alright, and comparative anatomy is we're comparing them, those structures between different species. Alright, so I've underlined a few concepts here which I want you to kind of make sure that you might want to kind of jot down some relatively simple ones. So what we're saying is that if you have parts of the body that are similar, this indicates descent from a common ancestor. It shows that evolution is a remodeling process where ancestral structures became modified as they take on new functions, and this is this whole idea of descent modification. When we look at comparative anatomy, we look at three different types of comparisons. We might be looking at homologous structures. So homologous structures are those with similarities. So if you look inside of an organism at the bone structure, you might find they're quite similar, but then when you look at the organisms themselves and their environments, those bone structures might do very different jobs. So a homologous structure is same or similar structure, different function. Conversely to that, you have analogous structures, where you have a similar function. I need to replace that. It's a similar function, but no shared evolutionary origin. And then we have vestigial structures, and these are structures that are no longer used. And a couple of examples of these are the tailbone in humans, hind leg and foot bones in whales, and the pelvis in snakes. So I'm going to go through a couple of examples with some pictures in a moment for you guys to have a look at. So let's look at our homologous structures. We're going to look at this a little bit more in detail in class, but what we've got here are 5 similar bone structures: the humerus, the ulna, the radius, carpals, and metacarpals, or phalanges. And that's in a human. We've got the same bone structures in a cat. We've got the same bone structures in a whale. We've got the same bone structures in a bat. So we've got walking upright and being the limb of grabbing. We have a limb for walking. We have a limb for pushing water around, and then we have a limb for flying. So similar structures, different functions. Alright, analogous structures. It's the kind of opposite to homologous. So we've got same function but different structures. The structure of a moth's wing is very different to some sort of flying lizard, to a bird, and to a bat. Very different, but they're under the same pressures. The pressure is, it's an adaptive advantage to fly, and so multiple organisms, if they live in a similar environment, will evolve similar structures to perform the same function. Okay, so looking at vestigial structures, these are structures that no longer serve a function now, but once they did. So whales have a femur, which is, in humans, it's the largest bone in the body. That theirs is this tiny shriveled up little thing that doesn't have a flipper attached to it. It's just sitting there floating in the tissue of the whale at the back there. We have hair on our chests. We have our wisdom teeth. Right? These are remnants from our grazing days. Right? We've also got our canines. And our ability to rip meat off the bone is not one that we particularly need right now, so there we call that a vestigial structure. We also have, above our ears, we have superior auricular muscle, and these help direct the movement of our ears. So some people can wiggle their ears, these muscles are slightly more developed, but it was largely from way back when our common ancestor needed the ability to move those ears around to either detect prey or avoid predators. We also have a very small appendix, a shriveled up appendix, and a tiny cecum. We aren't digesting grass material, and so that our human forms don't need to digest grass, so we've lost the ability to digest it. We don't have the organs for it. We have the organs, but they're shriveled up and they're kind of no longer doing that function anymore. Right, so we've got our tailbone. We don't need a tailbone. We don't have tails unless you go Super Saiyan, but you got nothing there. And then we have, I can never say this right, the necessitating membrane. I think I got it then. And it's that tiny little bit in the middle of in the center of your eye. Not center of your eye, the corner of your eye, and there's a membrane that goes over our primate ancestors or our primate cousins. They use it quite a bit to help clean their eyeballs, but we don't need it in humans. Alright, so there's a good example of vestigial structures that are no longer needed to serve a purpose. Alright, moving on to comparative embryology. Comparative embryology is the comparison of early stages of development before birth. So, we know that organisms share certain structures in different embryonic stages. And an example of this is the pharyngeal pouches in vertebrates. So this is kind of like the lung pouches, I guess. So comparative embryology is teaching us that similarities in embryonic development indicate organisms all evolved from a common ancestor. This idea of a common ancestor is going to keep recurring with all these things. It's a great bit of support to lend to evolution. So let's have a look at these pharyngeal. These are embryos, by the way. So we've got our human embryo and we've got our chick embryo. So we have pharyngeal pouches here, and then we've got our tail, which is past the anal cavity. So it will shrink and be used for other things in as the embryo develops. But early on, they have this post-anal cavity tail. Alright, so I'm going to go through something that should kind of demonstrate to you why embryology is important in this next slide. So have a think, have a gander, and hopefully it'll make sense. Across the top here we have the embryos of a variety of different species. This is early embryonic development, and you can clearly see they look similar. Yes, there might be some differences, but there are lots of similarities. As those embryos develop, though, genes either switch on or switch off, and the organism starts specializing in its structures, and we start to see that there are some differences. So over here, these are clearly different to some of these over here, and even this one's slightly different to this one over here. So if we look at the final embryo stage, you should get a bit of a shock to see just how different these species are. So we have a fish, a salamander, a tortoise, a chick, a hog, a calf, a rabbit, and a human, all originating from very similar embryos. Hopefully that really kind of reinforces why it was useful to look at embryos and compare them. I think it's kind of, I think, especially if we look at, there's a couple of better pictures of this. I'll find one in a textbook that's pretty good. But if you draw the line between endothermic and exothermic organisms here, so you just kind of these three to this side, the fish, salamander, and tortoise, share much more similar structures than these guys over here. And you can also see, remember how we're talking about evolutionary relationships, we can see that it makes sense that a chicken evolved from some sort of reptile at some point. I mean, just look how similar these are. It's pretty crazy. Alright, so that is comparative embryology. Last but not least, it is biochemistry, a relatively new field of study compared to these, compared to comparative anatomy or something. This is a very a new study. Sorry. An example of this is looking at the amino acid sequences and nucleic acid in organisms, and if there are similarities, this indicates a concept called relatedness. So the more closely the matches are in a gene sequence, the more likely that organism is evolved from a common ancestor. And a great example of this is looking at the polypeptide sequence of amino acids in hemoglobin. So if we compare humans to humans, there are no differences in hemoglobin. But if we compare humans to a rhesus monkey, there are 8 differences in the sequencing. And so because it's a relatively short polypeptide chain, which is only 146, this kind of is pretty cool for us. So with a mouse, there are 27 differences, with a chicken, 45, with a frog, 67, and with a lamprey eel, 125. So this suggests that the lamprey is the of these examples. The lamprey is the furthest common ancestor. We share it, the furthest back in time common ancestor with a lamprey eel. Hopefully that makes sense. Alright, and also with biochemistry, we have to kind of tip our hat to DNA and RNA. So we have found in all living organisms the use of the molecules DNA and RNA. It is everywhere. No life has formed that we know of that uses some other coding mechanism other than DNA or RNA. And this is a kind of a really big deal because it goes back to kind of like the origin of life, and then from that point onwards, the evolution of life. So it really says that all life forms are distantly related, even if they're ridiculously diversified now. Alright. Here we have the enormous summary. Hopefully that is useful to you guys. We have explicit classroom instructions classroom activities to help you guys understand this stuff a little bit more. But in terms of notes, you might want to get this down. Please note the language that we're using: homologous, analogous, vestigial, common ancestry, relatedness, missing links, this type of stuff. These are the type of language that you want to be using in any type of response that you might have in an exam. So get used to using that technical language like that. Okay, have fun. See you in class.